The overall goal of this procedure is to obtain enriched mouse embryonic motoneuron culture. The procedure begins with first sacrificing a pregnant mouse, removing the embryos, and dissecting the lumbar spinal cord. The next step is to dissociate the spinal cord into a single-cell suspension by enzymatic treatment and trituration. Following this, perform the lectin-based purification for separating the motoneurons from the cellular remainders. Next, after estimating the total cell number, the cells are plated on pre-coated dishes or cover slips. The final step is to adjust the density of the culture depending on the experimental procedure. Ultimately, as the cultured cells grow, typical motoneuron morphology can be observed.
For the success in preparing and culturing mouse embryonic motoneurons, it is essential to test different materials, substances and solutions, as the cells may react sensitively to any of these in changes.
The motoneurons can be cultured in high density for the isolation of RNA or proteins. However, there is a possibility that the cells will build up active neuronal nets which lead to the survival independent of trophic support. Therefore, in some cases, it might be better to culture the cells in low density for RNA or protein analysis. Demonstrating this procedure will be Rebecca Conrad and Teresa Chepan. Both are graduate students in our laboratory.
Begin this procedure by preparing the working solution of 0.5 mg per milliliter poly-D-L-ornithine hydrobromide in 150 millimolar borate buffer with pH 8.35 and sterilize the solution by filtration. Next, sterilize the glass cover slips by flaming them in 100% ethanol. Then, let them air dry. Cover the surface of the cover slips with sufficient porn H solution overnight at 4 degrees Celsius. On the next day, wash them 3 times with sterilized water. Then, let them air dry. After that, prepare the working solution of 2.5 micrograms per milliliter laminin in HBSS. Cover the surface of the porn H-coated cover slips with the laminin solution. Then, incubate them for at least 2 hours at room temperature until use. The next step is to prepare a solution of 10 millimolar tris in sterilized water with pH 9.5. After filtering the solution, add lectin to the mixture to a final concentration of 10 micrograms per milliliter. Then, coat the surface of a 10 cm cell culture dish with 8 milliliters of the lectin solution and incubate it for at least 30 minutes at room temperature. Next, wash the plate with HBSS 3 times and store the plate in HBSS until use. After that, prepare the depolarization solution with 30 millimolar potassium chloride and 0.8% sodium chloride. Then, sterilize the mixture by filtration and store it at room temperature. To begin this procedure, thaw horse serum overnight at 4 degrees Celsius. Then, inactivate it at 55 degrees Celsius for 30 minutes. Aliquot it into 5 milliliter portions and store the aliquots at -20 degrees Celsius. Thaw 1 aliquot directly at room temperature before use. Next, store B27 supplement in 1 milliliter aliquots at -20 degrees Celsius. Thaw it at room temperature immediately before use. Avoid the freeze and thaw cycles. Aliquot glutamax into 0.5 milliliter aliquots and store them at -20 degrees Celsius. Then, prepare a stock solution of 10 micrograms per milliliter CNTF in sterilized water with 0.1% BSA and store it at -20 degrees Celsius. Finally, mix 46 milliliters of neurobasal medium with 2.5 milliliters of horse serum, 1 milliliter of B27 supplement, and 0.5 milliliters of glutamax directly. Add CNTF to a final concentration of the medium of 10 nanograms per milliliter. Pre-warm it to 37 degrees Celsius. At this point, sacrifice an embryonic 12.5 pregnant mouse and remove the embryos carefully. Add room temperature HBSS to completely cover the embryos and move an embryo under a microscope. Then, remove the head and tail and place the embryo backside up with straddled limbs. Next, fix the embryo with one pair of forceps and remove the outer skin with the other pair of forceps. After that, Remove the spinal cord by piercing the forceps under it and lift it up with saw-like movements on both sides of the spinal cord. Transfer the isolated spinal cord to a new dish with HBSS. Open the central channel of the spinal cord on the dorsal side. Next, remove the dorsal root ganglia by removing the enclosed meninges of the spinal cord. Then, transfer the spinal cords to an Eppendorf reaction tube filled with 1 milliliter of HBSS. Keep them on ice until the preparation is done. For spinal cord digestion, transfer the tube with the spinal cords to a cell culture hood. Carefully remove 700 microliters of the HBSS. Next, add 7.5 microliters of 1% thawed trypsin solution, which was prepared earlier as described in the accompanying manuscript, and mix them by carefully inverting the tube. Perform trypsinization for 8 minutes at 37 degrees Celsius. Then, stop the reaction by adding 30 microliters of trypsin inhibitor. Pipette up and down carefully 10 to 15 times with a 1000 microliter pipette tip until no more cell aggregates are visible. After that, repeat the trituration with a 20 to 200 microliter pipette. Pipette the cell solution into the HBSS-filled lectin plate and disperse the cells by gently rotating the plate. Cover the lectin plate to avoid contamination and keep it on a vibration-free surface at room temperature for 60 minutes. Very gently remove the HBSS and wash the plate 4 times carefully with pre-warmed HBSS to remove cell fragments and unattached cells. Immediately after the last washing step, add 500 microliters of the 30 millimolar potassium chloride 0.8% sodium chloride depolarization solution to the plate and incubate it for 1 minute. Shake and tap the plate to facilitate the detachment of the lectin-bound cells. Fill up the plate with 2 milliliters of pre-warmed culture medium. Then transfer it to a 15 milliliter Falcon tube. Count the cell number with a Neubauer counting chamber. Next, plate the cells on the laminin-coated cover slips or plates. Change the medium on day 1 and subsequently once every 2 days by carefully replacing 50% of the old medium with pre-warmed, freshly prepared culture medium. Shown here is a cross-section of an embryonic 12.5 lumbar spinal cord labeled with Höchst for the localization of the cell nuclei. The arrow indicates the motoneuron columns within the embryonic lumbar spinal cord labeled with islet 1,2 antibody. An example of islet 1,2 labeled dissociated lumbar spinal cord cells from embryonic 12.5 embryos before and after lectin-based preplating are shown at the left and right panel respectively. The quantitative results of islet 1,2 positive cells before and after lectin-based preplating are shown here. Here is an example to show the enriched motoneurons on day 0 and day 2. Cells were fixed with 4% paraformaldehyde and subsequently stained for P75NTR according to standard procedures. They were then counterstained using Höchst to visualize all nuclei. After 5 days in vitro in porn-H and laminin as culture substrates and in the presence of CNTF for beta-3 tubulin staining, the cells have grown out long neurites and displayed the typical motoneuron morphology with 1 longer process and 1 or more shorter processes.
Motoneurons play a central role in the body of all vertebrates and invertebrates. This isolation method is extremely useful for the analysis of survival mechanisms of motoneurons. With the isolated cells, you can selectively analyze the activities of other interacting cells, proteins, carbohydrates, and apply pharmacological substances on the survival and neurite growth of these cells.